Gills are what fish, amphibians, and some other animals use to breathe in water. They are a respiratory organ that extracts dissolved oxygen from water and excretes carbon dioxide. Many microscopic aquatic animals, and some larger but inactive ones, can absorb sufficient oxygen through the entire surface of their bodies, and so can respire adequately without gills. However, more complex or more active aquatic organisms usually require one or more gills. Many invertebrates, and even amphibians, use both the body surface and gills for gaseous exchange. Gills have evolved to come in various shapes and sizes, depending on the animal or even the species. In many types of fish, there is a bony plate protecting the gills. This is called the operculum, which is hinged and has muscles attached to it, so that it can be regularly opened and closed. Underneath the operculum lies the gills. To prevent any large pieces of material making their way through to or damaging the gills, they have what's called gill rakers. This comb will catch any foreign bodies before they hit the gills. Each gill has a gill arch, and each arch supports one set of paired gill filaments. Each pair of gill filaments in turn supports numerous lamellae. The lamellae are collections of blood vessels sandwiched between two sets of membranes. The membranes keep the blood and the water separate, whilst allowing the dissolved gases, O2 and CO2, to diffuse naturally from the water to the blood and vice versa. The oxygenated blood is carried around the body, and blood containing CO2 circulates back to the gills, where the CO2 is expelled back into the water. The gills of vertebrates typically develop in the walls of the pharynx, along a series of gill slits opening to the exterior. When a fish breathes, it draws in a mouthful of water at regular intervals. Then it draws the sides of its throat together, forcing the water through the gill openings, so it passes over the gills to the outside. Most sharks rely on ram ventilation, forcing water into the mouth and over the gills by rapidly swimming forward. In slow-moving or bottom-dwelling species, especially among skates and rays, the spiracle, which is a small opening that lies in the back of the first gill slit in some rays and sharks, may be enlarged, and the fish breathes by sucking water through this opening instead of through the mouth. Tadpoles of amphibians have from 3 to 5 gill slits that do not contain actual gills. Instead of internal gills, they develop 3 feathery external gills that grow from the outer surface of the gill arches. Sometimes adults retain these, but they usually disappear at metamorphosis. Many marine invertebrates, such as bivalve mollusks, are filter feeders. A current of water is maintained through the gills for gas exchange, and food particles are filtered out at the same time. Crustaceans, mollusks, and some aquatic insects have tufted gills or plate-like structures on the surface of their bodies. Aquatic arthropods usually have gills, which are in most cases modified appendages. In some crustaceans, these are exposed directly to the water, while in others, they are protected inside a gill chamber, in which they store water, enabling them to use the dissolved oxygen when they are on land. A plastron is a type of structural adaptation occurring among some aquatic arthropods, primarily insects. It is a form of inorganic gill which holds a thin film of atmospheric oxygen in an area with small openings called spiracles. The trapped air of the plastron allows the arthropod to perform a gas exchange, almost as if it were in atmospheric air. This inorganic gill mechanism allows the insect to remain submerged underwater.